The overall goal of this procedure is to quantify the temporal organization, kinematic structure, and topographical features of idiosyncratic reach-to-grasp movements.
This method can be used to answer key questions related to the neurobehavioral organization of hand movements in infants, brain-injured patients, and in non-human primates who can be difficult to study using automated motion tracking techniques. The main advantage of this technique is that it is highly reliable, inexpensive, and unintrusive, yet it is easily modified to suit a number of research goals. Demonstrating the procedure today will be Alexis Wilson and Marissa Bertoli, who are research assistants in my laboratory.
Begin by selecting blueberries, donut balls, and orange slices to serve as reaching targets. Escort the participant into the testing room and inform them that they will be completing a total of 60 reaching trials separated into 4 blocks, with each block consisting of 15 reaching trials. Next, adjust the height of a self-standing, height-adjustable pedestal to the seated participant's trunk length so that the top of the pedestal stands midway between the top of the participant's hip and the participant's sternum. Then, position one high-speed video camera sagittal to the participant, on the same side as the participant's non-dominant hand, at a 1 meter distance from the pedestal, to record a reach side view of the participant's dominant hand. Following that, place a second video camera 1 meter in front of the pedestal to capture a front-on view of the participant.